Welcome to today's exciting webinar. Before we start, just would like to let you know the webinar is being recorded. So we will share it with everyone after the webinar. The webinar topic today is microplate agitation in cell culture and how C.NEST, C.BIRD unlock AI-enhanced bioprocessing and therapeutics. And the webinar is hosted by C.NEST BPS and Biocore Technologies. We are so glad you could join us today to explore how AI-driven microplate agitation systems are reshaping the future of the bioprocessing development. And I'm Charles, the CEO and the founder of C.NEST BPS, and I'll be your moderator for this session. And C.NEST technology is actually based on my own research when I was studying my PhD in Freiburg University, Germany. And I'm just so glad to see this technology now being used by pharmaceutical companies, research institutes, and other cutting-edge organizations in the field. Okay, and let's get started by meeting today's speaker. The first one, our first speaker is Dr. Joshua Hinckley. Dr. Hinckley has a unique blend of specialized in material chemistry, bioengineering, and AI-driven research. With a PhD from Cornell University and postdoctoral fellowship at MIT and also the Broad Institute, his work focused on the AI-assisted microbiota strategies, particularly in the blood cancer research. He's passionate about tackling complex challenges in biomanufacturing, and today he will share how AI is enhancing bioprocessing efficiency. And we are also joined by Ann Tsai, the product marketing manager of C.NEST BPS. With a strong background in the biotech marketing and bridges the gap between scientific innovation and practical applications. She plays a key role in positioning advanced technologies like C.NEST and C.BIRD to help researchers improve their workflows. She will be sharing how the microplate agitation technology is making a significant impact in the bioprocessing field. And we can take a look at today's agenda and follow by my introduction of the speakers. The second part will be Dr. Hinckley sharing his insight into how AI is transforming bioprocessing and the role of high-throughput reactor systems. In the third session, Anne will take us through the precise control, automation, and also parallel runs that make C.NEST microplate agitation systems stand out in the bioprocessing. And Dr. Hinckley will then share how Biocore's approach are supporting bioprocessing applications with a case study of combining the strengths of C.NEST and AI leads to time-saving, cost reductions, and a competitive edge in the fourth and fifth session. In the end, we will have a Q&A session to discuss the next phase and also to provide you with the necessary contact information to continue the conversation today. And yeah, before we dive in, just a few quick housekeeping notes here. The session will last about 1 hour, and we will be leaving some time at the end of the Q&A session. And so please feel free to submit your question through the Q&A button. You can see in the top row, there's a Q&A icons there. So you can type your questions there, and we will address as many as we can in the end of the session. Okay, now without further delay, let's keep things of Dr. Hinckley. The stage is yours.
All right. Okay, so good morning, good afternoon, good evening, wherever you're calling from. My name is Josh Hinckley. I'm the CEO and co-founder of Biocore Technologies, and we specialize in applying AI to solve complex, multidimensional, and nonlinear issues in bioprocessing. Next, please. So let's begin the discussion today about talking about the limitations of traditional methodologies. Standard methods are things like full factorial, fractional factorial, BDD, CCD, OFAT, all these like terminologies. They require significant planning and experience. They can also be expensive and time-consuming, especially when dealing with a large number of factors. As someone who's worked in this field, an automated analysis method that requires less than 30 experiments or less would be very ideal. So a core challenge in bioprocessing is this data bottleneck issue. So accurate predictions with AI or effective optimization heavily rely on high quality data. However, bioprocess data is often noisy, variable, incomplete, and gathering enough of this data can be very inefficient and very slow. Also very expensive. So this insufficiency leads to inaccurate model predictions and suboptimal optimizations. Next, please. So one AI method that Biocore utilizes is active learning, which addresses these data challenges. In active learning, the AI designs experiments, learns from the results, and refines the approach to focus on the most valuable data. This process leads to more targeted experiments and reduces the number of trials needed to achieve good results. Furthermore, this method is ideal for new product formulations or research on rare diseases or any other limited field by lacking prior data. So in bioprocessing, the need for speed is critical. There's an immense time to market pressure, but traditional methods are often really slow. So new technologies like AI-driven solutions are essential for accelerating innovation and meeting the demands of modern bioprocessing. So in the next sections of the webinar, we'll showcase more details about both of our technologies, C.NEST BPS's C.NEST system, as well as some of their other technologies, and Biocore's Voyager AI Manufacturing Copilot AI. And we'll explain how our collaboration is specifically designed to solve these challenges. And back to you, Anne.
Thank you. Thank you, Josh. Thank you, Charles, as well. So I'm Anne, the product marketing manager at C.NEST BPS, and I think today I'll be here to introduce to you guys the capabilities that our system can do, and where we call it the microplate agitation culture system. So a little background on what we've already achieved. I want to introduce to you guys this first. So we have been focusing on many different applications. The first is cell line development, and a lot of pharma company are already using our system in here. For example, Lonza or C.BIRD in Ireland. And then we also have customer culturing primary cell culture. That will be the example will be Broad Institute or MIT. And then some of the customer also culture spheroid or organoid. And one example data will be shown later, which is produced by the Ahead Bio in Austria. And we also focus on the application with the metabolism analysis with DO and pH readouts through the microplate education culture system. And also another one that is a relatively new field, which is the culture meat. I believe a lot of the audience also come from this application as well. So we can take a look into how our system can help you. So here is our system, C.NEST. We call it the microplate agitation culture system. So it's robust, it's flexible, and most importantly, is high throughput. So from the name of it, you can already get a few idea that we can do agitation in the microplate scale, 96 or 24 well plate, and it's doing the culturing at the same time. So this is what the device look like, and it's a benchtop device. And you can see that there are 4 doors at the front, and all of this is a incubator. So the incubator look like this. So it just function as the normal incubator that you know. So we can do temperature control, CO2 level control, humidity monitoring, and then we also add in another feature, which is the UV light sterilization for each chamber. The highlight I would put in here is that all these 4 chambers are independently controlled, so which means that you can have 4 different settings running at the same time. So 4 different and independent experiments running at the same time. Okay, and then it's what happened within each chamber. So each chamber, you will find 1 commercial plate, either 96 or 24 well plates, and then a C.NEST BPS patented lid on top. So the purpose of this lid is to generate the mixing for the well plates. So there are two little imports right here that will introduce the airflow. And then within the system, it will cause repeated suction and expulsion. And the system will modulate the air pressure through the import here to do the suction and expulsion. So which means that by modulating the air pressure, we can actually adjust the mixing, the agitation speed for you. So if you take a look at what actually happened in the wells, it will kind of look like this. So there is the tube of the lid, and then here is the well plate. So the liquid will go up and then go down to do the agitation. So as I said, we can modulate the air pressure so we can adjust the mixing speed. So for example, the slowest we can do is 50 seconds, and that means that the liquid will take 25 seconds to go up and then another 25 seconds to go down. And then if you want a faster mixing speed, for example, the 10 seconds one, it's quite a popular mixing speed for the chill cell world. That's for example. And then that means that the liquid would take 5 seconds to go up and then another 5 seconds to go down. So through this mixing technology that we are able to do agitation culture within the 96 and 24 well plate. And then just by doing that, we can create a higher oxygen transfer rate and also provide a homogeneous nutrition distribution for the cell culture. So in short words, that means we are providing a healthier environment for your cell to grow in. Okay, so since we are designing our own patented lid, and we thought that why not just solve also another issue, which is the corner and edge effect. So with the lid, we can actually control the evaporation. So in 96 and 24 well plate, we all did research on how the evaporation performed in our lid plate. So you can see that we put this into different region. So we have the corner, the edge, the inner, and the center. And no matter in 96 or 24 well plate, you can see that the remaining liquid volume are nearly the same in every region. So which means that our lid can also not only just to do the mixing, it can also help to minimize the corner and edge effect. And the benefit of this is that it's even giving you higher throughput, which means that you don't have to sacrifice the edge well. And you can use every well in your well plate, and you can even get more accurate data from here. Okay. And then I want to talk about the throughput in our system. So as I said, each chamber can fit in one set of the 24 well plate and or a set of 96 well plate for the mixing culture. And then if you do some calculation, you put 96 well plate in all 4 chambers, then that will give you 384 clones, or let's say 384 different conditions at the same time in one system. And because the chamber is also an incubator, so also some of the users use that as just an incubator, so to do the static culture. And if you're going for static culture, then you can actually stack the plates. So that means one chamber can fit in 124 well plate, or you can stack 296 or 384. And that maximum will give you more than 3K of the condition, or 3K of, more than 3K of the clones. Okay, and then here is what our software will look like. So it's actually very user-friendly. So right here, you will see 4 different chamber status shown in here. And in the animation here, you can see the mixing figure. So you will see it go up and down along with your mixing. And then you can also see the incubation environmental data, for example, how much percentage of the CO2 you're using and the real-time data of it, and also the temperature, the humidity, it will all be shown here. So it's quite simple, quite straightforward, and very easy to get on hand. Okay, and since our customer are culturing many different types of cells. So controlling and maintenance about the sterilization is one point that customer will care about. So there is another design in our system that the plate holder that is designed to hold the plate and the lid, it's actually removable, that you can remove it from the chamber. So it will simplify the cleaning for all the users. So it's better efficiency if you want to maintain the incubation environment. And also, once you take out the plate holder, you can actually take that to autoclaving. So that is just to maximize the sterility, and for repeated use, or even many users is using the same system, it's better to keep it that way. And then it's about water tray. So as I listed here, we have a disposable water tray. Just like the normal incubator, we also designed the water tray component here, but we design it as a disposable one. So there is no worries about any carryover of the contamination, because you can just replace one in between each experiment. Okay, and then another feature of C.NEST is that you can actually maximize the throughput even more. So one system has 4 chambers, so 4 independent experiments at the same time. But also our system, the software can also control 4 systems at the same time. So that means you will connect 4 C.NEST into one laptop and all controlled by one software. So that would give you a total of 16 chamber that is interference free, that you can run 16 experiments at the same time. Okay, and that's the overview of C.NEST. And I know we have another start here, which is C.BIRD, that is also mentioned in the topic of today's webinar. So a little introduction on C.BIRD here. So as you can see, it looks a bit simpler than the C.NEST, and you can understand C.BIRD as just like what C.NEST can do. It will also do the mixing for 96 and also 24 well plate, but the difference is that it doesn't have the incubation function, so you will have to put the entire system into your incubator. But the benefit of this is that you can actually carry C.BIRD from one lab to another. So a lot of people in your institute can actually try out the system by simply just plugging and plug out the C.BIRD and bring it over. It's very simple to install, and it just comes with a laptop to control it. Okay, next I want to introduce some of the featured application. So first is the cell line development. In this application, we actually been putting a lot of effort in it already, and already see some success story. So here is our proposed workflow. As I said, we already put a lot of effort in this application, so we actually can propose a more modern workflow for the CLD field. So in this world, the CLD workflow, cell line development workflow, always have to go through transfection and then single cell seeding or nanopen technology, and then go through a long process of scaling up from, for example, 384 well plate, and then 96, 24, and then 12, and then 6, and all the way up to maybe tube spin or shake flask. And then what C.NEST and C.BIRD can do here is to introduce the mixing technology at 96 and 24 well plate format. So which means that it's already providing a better cell culture environment at this time point. And then that will brings you a higher cell number at this time point. So maybe you can skip the 12 and 6 well plate culturing and start the tube spin or shake flask at an earlier time point. So that is saving time for you and also saving cost of all the other medium and all the other reagent. Okay, and then here is a more detailed comparison of the workflow. Up here is the conventional workflow. So as I said, it's always static culture from a small scale to a bigger scale. So for example, 384 well plate or 96, and then all the way to 24 or 12 or 6 well plate. And then I put a little market research data here that a lot of the scientists actually thinks that this process is taking too long. It's normally taking 3 to 4 weeks or even more than 4 weeks. So that's the pain point for a lot of scientists here. And then what we can propose is to still start with the static culture for the clones to be more stable. And then we can initiate and introduce the agitation culture, the mixing culture from 96 and 24 well plate. And from our database where we co-work with a lot of customer and also collect the system performance from them, we found out we can actually save 29 to 42% of the time for them. And here is actually a one customer data that we recently collected that it's performing really well. That's saving 42% of the time for the customer. So in this example here, their conventional workflow is start with 96 well plate static and then move to 24 well plate after 15 days. And then this is the time point where they start with the shaking. And then overall, it will take them 31 days before the cells can go into the tube spin. But once they introduce the mixing from the 96 and the 24 well plate, they can greatly shorten the time from 31 days to 18 days. And that is 42% of the time. So it's quite phenomenal for them, and it's proven very helpful for their workflow. Okay, and here is just some of the testimonial that we collected from the customer. But I guess the takeaway message here is that introducing the mixing technology at early stage in 96 and 24 well plate can greatly introduce a better mixing, better cell culture environment at a small scale, and also bring in the cell number up at the small scale. So mainly helping the customer with saving time and also labor and also the cost of it. Okay, and then it's the second application that I want to introduce today, which is the organoid culture. So one famous example that we have here is a customer that was based in Austria, and they are culturing iPSC-derived brain organoid. And the bottleneck they have is that they want a higher throughput system to culture their organoid. And the conventional workflow they have is the 10 centimeter dish, but they want to culture it in a 24 well plate format. So they tried out the static workflow and also the C.BIRD mixing workflow. So they culture it for a very long time and comparing the size of the organoid through the imager. So as you can see, if we directly take a look at the image on day 39, you can see that the organoid is growing much bigger than the static culture that they used to have. And then, of course, organoid is just not about growing bigger. They also want to have the correct morphology that they want. So they also do a calculation of that, and you can see three comparison group. One is their the lower throughput way, the conventional way, is the 10 centimeter dish, and then we have the pink group with the 24 well plate C.BIRD, and then the static group with 24 well plate. So with their calculation, they can actually find that the C.BIRD mixing is not only giving them a bigger growing organoid, but also giving them the correct morphology, the correct budding of the organoid. Okay, these are the examples that I want to use today's webinar to share with all of you. But of course, we have many other data and many other cell types that we want to share with you, but maybe not to include in today's webinar. But you guys can always contact us to see more. So I want to end my section with a quick comparison chart of the product we have. So first is C.BIRD. That is a relatively simple mixing instrument that has to be placed in the incubator because it doesn't come with the incubation function. And then the throughput is 3 well plate in one system, either 24 or 96 well plate. And then we have C.NEST that has the mixing function and also the incubation function. And the throughput is slightly higher, that it has 4 chambers, so it can do 4 experiments, either 24 or 96 well plate at the same time. And then you can also use it just as an incubator for the static culture. Okay, and then another product that is probably not the star today, which is C.NEST, that I'll just quickly blow through the capability that C.NEST has. That you can understand C.NEST as C.NEST, but it also has the DO and pH monitoring function. So this is what it looks like. So it will also have the mixing in the well plate, but with the well plate that it comes with, it will have the dissolved oxygen sensor tag and also pH level sensor tag mounted at the bottom of the well plate. And it will give you a real-time monitoring of the DO, pH, and OCR, ECAR at the during your cell culturing. And then the actual data will kind of look like this, that this is a very simple, quick data just to seed it with different seeding density of the cells. And for example, you can see that if we take a look at the green group, that the DO and the pH is dropping the fastest because it has the highest seeding density. So the oxygen consumption is dropping the fastest, and also the pH is dropping the fastest. So the highlight in C.NEST is just that it does the mixing and also DO/pH sensing during the cell culture period, and also it comes with the incubation function. So it's a long-term system for the mixing and also the sensing. Okay, and just to end here, that this is all our product, C.NEST, C.NEST, and C.BIRD, but the main takeaway is that we all can do 96 and 24 well plate, microplate agitation culture system, so providing you with a higher throughput system to do the culturing and also the analysis that you need. Okay, and then I guess I will leave the next session to Josh.
All right. Okay, to start our section for BioCore, I'm going to start with talking about why active learning. Go ahead and hit the next slide, please. Okay, so our approach is active learning, which provides faster insights and fewer experiments with more efficient resource utilization and more intelligent design, making this very much a high competitor, or even better than traditional methods or random sampling methods. This is very ideal for these high complex multidimensional issues that it may take a long time to optimize a proper media, and it can take over 20 different factors inside of that media. We can handle that problem, no problem. Next slide, please. So our Voyager AI powers this experiment design by itself. So it considers multiple parameters and their interactions for a comprehensive analysis, and it optimizes for information and efficiency, and also enables rapid exploration of design space to identify optimal conditions. And I'll talk about this animation in 1 or 2 additional slides. Next, please. So this is our process. This is our active learning refinement process. So it begins with the user defining variables of their experiment, such as concentration, taste, if you're working with some alternative meat products or milks, and etc., temperature. These variables are fed into the Voyager agent, which predicts initial sets of tests that need to be performed. Following these tests, the user gathers all the information and feeds it back into the AI agent to train the model. This process kind of continues over a session of 1 or 2 additional rounds. And then if it determines that an ideal combination of factors have been found that meet the objective, you've finished your experiment and you can go on to the next project. If not, it'll recommend doing another set. This iterative-based learning efficiently guides these users to achieve their desired outcome very efficiently. And so you might say, hey, it still takes 3 times, but 3 times versus, several weeks of experimentation, you have to kind of figure out which one is your thing you're willing to do. Next, please. So this is one of our experiments that we actually utilized the C.NEST platform during my postdoc. This is a example of a optimization of cell growth of cancer cells in culture media that we're trying to optimize the growth factors inside the media that provide the best cell growth possible. So in the first iteration, this is our initial space exploration phase. This is where the user will provide us with the conditions or initial boundary conditions, if you will, that we need to figure out what combination of these things works the best. In the second round, this is what we call the first exploration phase. This is where the machine learning algorithm determines that there's not enough information to accurately predict any outcomes. And this should be a GIF, but it's okay. You have to take my word for it. In the final round is the exploitation. This is where it takes all those former data points and determines the final outcome. And so from our experience, between 3 and 4 iterations, we can efficiently figure out what's the optimal combination of all these parameters that lead to your desired outcome. The bottom graph is what we use to compare the number of experiments for a process such as this compared to state-of-the-art DOEs. And you can see no matter how many different parameters that you add in for the Voyager, it basically looks like a linear line. Versus if you use these design of experiment methodologies, this type of scaling can basically is exponential. Next slide, please. So here are some of the very interesting applications that we're currently using Voyager for, from therapeutics to recombinant protein manufacturing, semiconductors, food, cosmetics, chemical processes, and even industrial coatings for cars and aerospace. And so Voyager can achieve these by, next slide, please. By some of our particular IP that I won't obviously go into a ton of detail, but there's 3 things that really contribute to our universality. First is our Voyager probe sampling. This also takes into account any previous knowledge the user has. It doesn't have to be data. It could just simply be, temperature influences growth. And that information we can use to actually prioritize some initial sampling based around temperature. We also have this adaptive probability kernel, which is kind of where the true power of our system lies, that no matter what type of process is being utilized, we can adapt our probability density functions to adapt. So what do I mean by that? So in the figure on the right side, the top shows a typical biological process. They typically follow a Gaussian type distribution. But when you look at nanoparticle synthesis or even chemical optimizations, these tend to be more discrete and periodic. And so if you were to traditionally come in with a normal non-Voyager type of companion, you might only be stuck with one of these type of processes. But for us, the probability, the adaptive probability kernel determines which probability is the best for your particular case. So it's nothing that the user actually has to define. And finally, we have the rapid reward optimizer, which allows us to train our models much more efficiently and get a better reward for much more fast optimizations, essentially. Next slide, please. So looking ahead, the future of bioprocessing is intelligent. This involves AI-driven automation, where intelligent AI systems increasingly automate bioprocess workflows. It also involves high-throughput AI synergy, where AI efficiently analyzes large data sets like systems like from C.NEST for predictive bioprocessing. And this also helps with our reducing of trial and error and provides us a more data-centric approach and effective data management, and where we want to make sure that all of our analysis is central to the kind of problems we're trying to answer. And next slide, please. And to end our section, so we are actively seeking collaborations to further apply this technology. We are actively looking for partners for initial validation through a variety of different applications in bioprocessing. This is a unique opportunity to apply our cutting-edge AI solution to your specific bioprocessing challenges and unlocks some very interesting results. We're also looking for investors to further accelerate our innovation and expand our impact across bioprocessing industry. So if you're interested with exploring a partnership or just want to chat with us, we're a fairly nice group of people. Please reach out to contact@biocore.ai or just go to our website and submit us a request. Okay, to finalize our webinar, so we discussed these challenges in bioprocess optimization and how AI can address them. But now let's explore how we can combine these two really cool technologies and to maximize efficiency. Next, please. Perfect. So our companies have teamed up to provide a streamlined solution to bioprocessing and using high throughput reactors and automated AI analysis for optimization. So our collaboration combines these really key areas of expertise in high throughput experimentation and instrumentation with intelligent experiment design. Next, please. The combination of C.NEST high throughput experimentation and Biocore's intelligent AI optimization empowers users to accelerate development, optimize processes, increase productivity, reduce costs, and make data-driven decisions. So this synergy enables faster innovation and brings products to market much more efficiently, providing a distinct advantage in the bioprocessing industry. Next slide, please. So to summarize, C.NEST and the Voyager AI work together to accelerate bioprocess development. The Voyager AI and C.NEST automate and accelerate these bioprocess optimizations, resulting in fewer experiments, faster results, and lower costs overall. So we'd love to collaborate with everyone.
Great. Thank you, Joshua, and also Anne here for the very nice presentation today. Just a very quick reminder to everyone that if you have any questions, please, there's an icon Q&A in the top row here, and you can type your question here, or afterwards, you can, contact us through LinkedIn. You can scan the QR code here, and we are happy to answer you offline as well.